Mendel's three principles. We want to take a quick second to review his three principles.
Carla Nava, please come back to room 710. Carla Nava, please come back to 710 immediately.
His three principles explain how some traits are inherited, and we call these patterns. So certain traits, like rolling your tongue, having a hitchhiker's thumb, having freckles, follow Mendelian principles. So Mendel's three principles, the first is the principle of dominance. That one version or allele of a gene is stronger than the other versions. But the weaker version, the recessive, is not missing, it's just hidden. So the concept that the recessive is hidden by the dominant. And so this would have been our P generation, this would have been our F1, which are all heterozygotes. And then here we see our 3-to-1 phenotype ratio, but we know that this is actually a 1-to-2-to-1 genotype ratio. So Mendel's three principles work when one version is stronger than the other. They also are going to work when we look at segregation. That each parent donates half. And so what we see here is the concept of donating and each parent donating half. Each parent contributing to the offspring. The third principle is the principle of independent assortment. And so it does not mean that the traits are, it does mean that the traits are inherited independently. If I have a green wrinkled pea, that doesn't mean the only way to be wrinkled is to also be green. I can be green and be round. I can be wrinkled and be yellow. And so this is the example of how your traits are inherited mostly independently of each other. So these are Mendel's three principles. But it turns out that more is going to contribute. There are going to be a lot of patterns that aren't as easily explained. So most organism traits are not as straightforward as the pea plant was. The pea plant traits were very straightforward inherited. They followed those principles that Mendel was able to observe. But in a lot of other organisms, we see different patterns, we see other contributing factors. So nature is your DNA, your genes, and nurture is your environment. And so it turns out that there's a couple things that come into play here. You probably remember the nature-nurture debate. We have some traits that are all environment, like the language you speak. We have some traits that are a combination, like your height. So your genes will determine your maximum height, but your nutrition determines whether you meet that maximum, same with your weight. But there's a really interesting phenomenon that's been discovered where sometimes your environment can permanently impact your genes. Even that are passed on to your offspring. And this is the study called epigenetics. And the evidence that was first found was when they were studying the offspring of people who lived through the potato famine. So it turns out that people who are raised in an environment where they don't have a lot of food, their genes are actually permanently changed due to how their body has to modify if there's not a lot of food. and the change to those genes is actually passed on to the offspring. So what they saw is that in the famine, you had a lot of really, really slender people. There wasn't a lot of food. Their bodies were making do with what they had. It turns out that the next generation was back to normal food supply, but all of a sudden they were very much obese because they were born with genes that were expecting a famine. And so that extra food was getting stored compared with a normal population that had had permanent availability of food. So we do kind of want to know this word. It actually turned up on the AP exam last year, so it probably won't be there this year. But we do want to know that there are things in the environment that affect the expression of a gene, so whether a gene is turned on or turned off, but also impact the sequence of the gene. What we're mainly going to be focusing on, and of course the best way to study this is with twins because they have identical DNA. The main thing that we're going to do is we're going to study different patterns of inheritance. Because for some traits, there is no way to know what's going to happen. So for example, with personality, part of it's genetic, part of it's your environment, but we really can't predict exactly what a child will be like. Other things we can find certain patterns. So we're going to be talking about looking at multiple traits at a time. So 2, we'll also look at more than that. We'll be looking at a phenomenon called codominance, incomplete dominance, multiple alleles, epistasis, pleiotropy, and polygenic. Oh, and I almost forgot a really important one, sex length. So today we're just going to look at these 3, dihybrid, co, and incomplete dominance. And then we'll be looking at the others on a later date. So what this means is that the product of the gene is going to follow different patterns. If the pattern is incomplete dominance, the product of each allele exists and gives us a mixture. So in dominant and recessive, a lot of times the product doesn't exist. The recessive is a mutation, it's not even made. Here, it's going to exist and the 2 are going to mix together. So the best example of incomplete dominance is 4 o'clock flowers. Now it's really easy to make up made-believe examples like a big cockroach and a little cockroach and their babies are all in the middle. What we want to do is try and keep ourselves grounded in real examples. So we're going to use the 4 o'clock flower a lot. 2 pink 4 o'clock flowers are crossed. Anytime that we see a mixture, like pink or gray, we want to identify that word I and look at incomplete. So incomplete dominance is where they're going to mix together. You can really write the letters however you want as long as you remember that the phenotype mixes. I like to follow the patterns that represent each pattern with a different type of letter. So in dominant and recessive, we use capital and lowercase. In incomplete, we will use 1 capital letter for 1 form. We'll use the same capital letter but with a prime symbol. I actually think of it as a little I. And that will equal the other form. The only way to get the mixture is to combine the 2. So, would be R, R. A white flower would have two white alleles, R prime, R prime. And when these have offspring, they each get a big R from one parent and an R prime from the other parent. And that's where I get the mixture. There is no allele for pink. Pink exists because red and white are mixing. So this is why it is really important to assign your alleles. If we work out this problem, two pink four-o'clock flowers are crossed. R, R prime times R, R prime. Now you might be able to do this in your head, or maybe you want to fill out the Punnett square. R, R prime, R, R prime, R, 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 R prime, R, R prime, R prime, R prime. And so the phenotype ratio, right, as for the genotype and phenotype ratios, the genotype ratio is 1 RR compared to 2 R prime compared to 1 R prime, R prime. The phenotype ratio, what is my physical appearance? 1 red compared to 2 pink compared to 1 white. Then it wants to know what is the chance the offspring will have the same phenotype as the parents. The parents were both pink. The chance of being pink is 2 out of 4, or 50%. And so this is incomplete dominance. The key point here is being able to recognize when it is a mixture, and then really make sure that you define your letters so that when you get to the end, you know what those letters actually mean. The other example is codominance. Codominance is going to be when the product of each allele is separate but present, or separate but equal. So a really good example is the checkered chicken and the roan cattle, R-O-A-N cattle. So again, we could say that like Dalmatians, white and black, but if you cross 2 Dalmatians, you don't get all white Dalmatians or all black Dalmatians. So obviously, in Dalmatians, spotting is not this example. But again, what we need to do is be able to look at a problem and then be able to make some predictions. They would really need to give you information so that you are sure of the pattern of inheritance here. But let's use a checkered chicken problem. We would give a capital letter for one version and another capital letter for the other. Now again, the types of letters you use, that's not going to be a test question. But you do need to know the relationship between the letters according to each pattern. So you can go around using big A and little a for everything, as long as you knew what the letters represented. I think that's confusing. So I like to follow the rules where we use different patterns of letters for different patterns of inheritance. For codominant, they are equal, black and white, not mixing to make gray. So the example here would be, for example, a black chicken, BB, times a white chicken, WW. All of the offspring would be black and white. And then we could cross that again. So this would be our P generation, our F1 and our F2. You can go ahead and work this problem as well. And then on Monday, we're also going to look at some dihybrid examples.